Okay, good afternoon. Good afternoon to all of you. And today, so in the last class, we looked at, northern blots, western blots, all the blotting techniques, So both southern blot and northern blot involves hybridization with nucleic acid, whereas western blot is an extension of that in the sense that instead of nucleic acids, we are looking at protein-protein interactions, right? Antigens in large part are proteins. Antibodies again are proteins. So we are looking at specific antigen-antibody interactions in Western blots, right? The principal actors are different, right? In the case of southern blot, it is going to be genomic DNA, and the probe that we are going to use is also genomic DNA or cDNA, fragment of cDNA, depends on what piece of probe we have, right? In the case of northern blot, you actually run RNA, right? You either run total RNA or you would run the purified messenger RNA on the gel, because messenger RNAs tend to have a long poly A tail in eukaryotes. You can very easily purify messenger RNAs by simply taking a cellular extract and passing it over what is called as either a poly T column or a poly U column, right? Generally, people prefer a series of DTs, oligo DT column, and T and A will have a specific base pairing, and so your messenger RNAs can be enriched without any hiccups, right? And then your transfer RNA and ribosomal RNA, which account for more than 90%, are removed. And so what you have in the fraction is basically essentially only messenger RNA, and messenger RNA can vary anywhere between 2 to 5%, right? So the amount of messenger RNA in a cell generally doesn't go beyond that, 2 to 5% maximum, okay? So it is easy, but only thing is RNA is extremely fragile, fragile in the sense it can be chewed up by nucleases just like that. Unfortunately or fortunately, the half-life of RNA, imagine, I mean, just compare between RNA and DNA. DNA is stable for generation after generation after generation, right? On the other hand, if you look at RNA in the cells, specifically messenger RNA, they have extremely short half-lives, right? They can range in the matter of few minutes to a couple of hours, typically in prokaryotes. In eukaryotes, certain messenger RNAs can be long-lived, but again, there is a range, so at some point of time, the messenger RNAs get degraded, right? And all this is catalyzed by an enzyme called ribonuclease, okay? Ribonuclease is one enzyme which you can boil and allow it to cool, it will regain its activity. Our classical biochemistry says that when you boil an enzyme, the enzyme will lose its activity, right? But there are exceptions, right? RNase A is one such example where you can boil the enzyme and allow it to cool, right? And when you allow it to cool, it refolds and regains its activity. May not be 100%, but it regains a large chunk of activity, which is of concern because somehow when you purify your RNA, we need to make sure that RNA is not getting degraded by ribonuclease. So researchers take extreme precautions in making sure that all the RNases are inactivated, right? They use a variety of techniques. The chief among them is like autoclaving and baking, baking at 80 degrees centigrade for a couple of hours will inactivate to a large extent these things. Use of gloves, our skin, our skin with bare hands, when you touch it, sweat is loaded with RNAs, okay? It can chew up RNA, right? So we don't want to be, whenever you're handling RNA, you will take extreme precautions. You will wash the glassware with certain specific chemicals. You will of course these days there is a lot of commercial solutions that are available that can inactivate RNA on the go, right? Immediately, right? Earlier, that was not the case. So people took a lot of precautions, and working with RNA, in any case, needs a lot of patience, perseverance, and then you will become adept, or you will become an expert in working with RNA. Okay, so when you come to northern blots, you would be running the either the total RNA or the purified messenger RNA on agarose gels. So both southern blot and northern blots are typically run on agarose gels. Only thing is, because RNA assumes this extensive secondary structure, in order to denature it, people generally use either formaldehyde, in the agarose, or they use some other few other chemicals to make sure that we have a denatured RNA. Okay, so the easiest way will be to have a formaldehyde agarose gel. You don't use that for southern. You will use formaldehyde-based agarose gels for northerns only. Okay, and when it comes to western blot, typically polyacrylamide gels are used, not agarose gels. Okay, polyacrylamide gels are used for separating the antigen, and in all these cases there are certain commonalities, there are certain differences, right? The buffers used are different for each of them. We simply use TA or TB as the case may be for southern. You will use additional formic acid for northern blots, and for western blots you will be using a totally different buffer. It is not TAE or TBE, it is a lamellae buffer, which is typically used for separating proteins. Okay. The commonalities include transferring it to nitrocellulose or nylon or PVDF membrane. So transferring it to membrane from the gel, could be agarose gel or acrylamide gel, is something that is common across all the formats. Detection again is something that is common, except that the way you detect can be different, right? So for in the case of southerns and northerns, you could use a variety of techniques. Okay, the probes are radio labeled with P32 or any other appropriate nucleotide, and then you can detect it by autoradiogram. Okay. In the case of western blots, you can do a very similar thing. Either you can have antibodies that are labeled with iodine 125, or these antibodies can be coupled to certain enzymes. Okay, and when you add substrates, if there is an antigen antibody interaction, you will have a color development. If the antigen is not found and the antibody doesn't bind, then you will not have any color development. Okay, very simple and very powerful techniques, which actually tell you, very useful in clinical diagnosis. For example, before the RT-PCR test became available, Western blot was the gold standard for HIV patients, right? So they had this very powerful antibody that could detect HIV, in the blood. And all that they will do is take the blood from an infected individual, few mLs, okay, and they will lyse open the cell, and then because your HIV would have made some proteins, these antibodies will detect the HIV protein even before the disease is manifested, right? So when they suspect that this could be HIV, they will do that, and then the patient is put on antiretroviral medications, and the patient has to be on that anti-HIV medication for lifelong. Okay. It turns out that Western blot was gold standard until a few years ago, when real-time PCR became the most preferred tool because of advancements in detection. And now you can detect HIV in the patient's blood sample. Even like as low as few copies of HIV can be detected. Okay. For Western blot, you need a substantial amount of infection to progress. Only then, when the doctor suspects this could be HIV, they do a Western blot for confirmation. Whereas nowadays, RT-PCR has become so powerful that you can detect the presence of few copies of HIV in the blood, and the patient goes for extensive and aggressive what is called as heart therapy, highly active antiretroviral therapy, right? And the disease burden is managed to a large extent in a normal fashion. Okay? So these are what happens in southern, northern, and western. Of course, southern blotting technique was developed by a scientist called Ed Southern, and so he named it as Southern blot, right? And just out of fun, somebody said, if Southern means DNA to DNA, can we call RNA DNA or RNA hybridization as Northern? And people just casually named it Northern blot, but for some reason it got stuck. The scientists who invented Northern blots were two different people. Okay. And similarly, when Southern and Northern became a norm, somebody said, why don't we call the antibody antigen interaction as Western blot? And so, somehow those names got stuck, those labels got stuck. And then I also said that there is something called as a Southwestern, which basically looks at things like DNA binding proteins, which can interact, with the DNA. For example, DNA polymerase. You have single strand DNA binding proteins, right? You have many different kinds of proteins, topoisomerases, right? Ligases. There are several enzymes that can bind to DNA. That binding to DNA is characterized by what is called as Southwestern, which we are not going to discuss at this point in time. Okay, but just for your this thing, I'm saying that there is also something called as a Southwestern blotting. Okay, so for today's class, what I thought was, we all know that human genome is 3 billion base pairs, right? 3 times 10 to the power 9. And if you remember the class on, when we talked about how to use this for DNA fingerprinting, the very reason why DNA fingerprinting became possible is because of what is called as polymorphisms. What is polymorphism? I'm sure you would have heard about this terminology called polymorphism in your senior school, which basically means the existence of many different forms of the same thing, right? So although all of us share 99.9% of the genome, about 0.1% varies, right? The 0.1% varies between any two individuals, except when they are homozygotic twins, right? So this 0.1% variation itself will be of the order of few million nucleotides. Now, in addition to that, we also see a lot of changes, okay? And these changes are what are called as single nucleotide polymorphisms, okay? And single nucleotide polymorphisms have a lot of relevance in human physiology, not only in human physiology, but also in other life forms, right? For example, when it comes to plants, when it comes to animals, these SNPs can have a very, very significant impact. Okay, so let's quickly look at what SNPs are, and how are they useful and all that. Okay. So any questions on southern, northern, and western blot? Okay. So is the screen visible? These are short videos, so hopefully we will have a clear idea.
Introduction, SNPs, significance of SNPs, types or classification of SNPs, methods of SNPs detection, applications, and its advantages and disadvantages. So let's get started with the introduction. SNPs are DNA based marker or molecular marker or genetic marker. So the question is, what is genetic marker? Just like a marker is used to indicate something, genetic marker in genome is that part of DNA sequence that marks a specific area and acts as a molecular marker to identify different individuals of same species. For example, in this picture, the green area shown in the DNA sequence is a gene sequence with two flags acting as genetic markers for that specific gene area. So this is called a genetic marker.
So you don't see a flag in the this thing, but what you see instead of flag is that you will see certain specific sequences. For example, in this case, you may see a promoter sequence somewhere here, and you will see a terminator sequence somewhere here. So specifically, when we talk about human genome, for example, we all know that there are lots of introns, lots of exons, and each gene will have its own promoter, will have its own terminator. So the structure, the genetic structure or the architecture of genes are very, very different, right? So the beginning of the gene, upstream of that, you will have a lot of promoter sequences, other regulatory sequences, right? It could be, enhancer sequences. It could be, regions for transcription factor binding sites, many different things. So that is what is called as some, you don't see a literal flag there, but the flag actually tells you that, there is something downstream or upstream of this, okay?
Genetic marker represent genetic difference. It is located close to genes. Other DNA-based markers are RFLP, RAPD, AFLP, SSR.
So these are RFLP, RAPD. These are the techniques that are used to detect for the presence of these kind of changes in the DNA sequence. Okay.
SNP, etc. Advantages of molecular markers are these are neutral sites, these are located in known coding sites, they don't affect the gene of interest, these are numerous and abundantly available, these are independent of environment.
So one thing that characterizes this SNPs are, if you look at the genome, it is 98% of the genome is non-coding, only 2% is coding. So the probability of SNPs occurring in the non-coding region is several orders greater than it happening in the coding region, right? Yet it does happen. Yet it does happen that you see some variation. That's why you have, these different eye colors, right? Eye color is a typical example of an SNP.
Environmental factors. Now let's discuss about SNPs. SNP stands for single nucleotide polymorphisms. SNPs are also pronounced as SNPs. Polymorphism means many forms. That means many forms of single nucleotide. SNPs are DNA sequence variations. In this picture, the, the DNA sequences of these three individuals differ from each other just because of a change in the base pair or nucleotide. Just a change in a single nucleotide. Hence, this is called SNP, single nucleotide polymorphism. Hence, it is a DNA sequence variation occurring when a single nucleotide A, T, G, or C in the genome differs between members of a species. These are most abundant source of DNA polymorphisms in organisms and act as genetic marker. SNPs were first described by Jordan and Humphreys in 1994. Another example we can see, for example, A individual has a sequence GAACCT, while individual B has sequence GAGCCT. So the polymorphism is AG. Causes for SNP can be due to the result of errors in replication, damage of DNA, transition, transversion, deletion, insertion, and significance of SNPs. SNPs are found mostly in non-coding regions. They are codominant, that is, distinguish between homozygous and heterozygous. 90% of variations in humans are due to SNPs. This could be harmless or harmful. Harmless in the sense changing phenotype, and harmful in the sense to cause diabetes, cancer, heart disease, etc. Types of SNPs. These are of two types: non-coding region SNPs of DNA, in which the SNPs are present in between genes, and in coding region SNPs of DNA, that means SNPs present in the gene. Coding region is further divided into non-synonymous and synonymous SNP. Synonymous SNP is a silent change. It occurs in the change of codon but no change of amino acid in the protein. Non-synonymous SNP is further divided into two: missense SNP and nonsense SNP. Missense SNP leads to the change of codon, results in a change of one amino acid in the protein, while nonsense SNP creates a stop codon.
So typically in a missense SNP, one aromatic amino acid could be replaced by another aromatic amino acid. For example, tryptophan could be replaced by phenylalanine or vice versa. Or, an aliphatic, an acidic amino acid gets replaced by another acidic one. For example, glutamic acid could be replaced by another acidic one, right? So things like that. By and large, these, although they are missense SNPs, the amino acid has been changed, but the by and large the function remains the same. Okay, unless when you have replaced an aromatic amino acid with another one, then it becomes a problem. Otherwise, by and large, these substitutions are tolerated, and that is why this is called as a missense SNP. Okay. Nonsense SNP, on the other hand, is going to create problem because it is going to create a premature stop codon
In the gene and results in premature truncation of the protein, that is incomplete protein made. Methods of SNP detection. SNP detection can be enzymatically based, electrophoretic based, solid phase based, or chromatographic based. Enzymatic based are RFLP, AFLP, CFLP, RAPD. Electrophoretic based single strand conformation polymorphism SSCP. DNA sequencing. Solid phase based oligonucleotide arrays. Chromatographic based, that is DHPLC, denaturing high performance liquid chromatography. Let's see one by one the detection techniques. Enzyme based SNP detection method. If the target SNP has site for restriction enzyme, then the PCR product of such SNP locus will produce a differential restriction platform. Example: RFLP, AFLP, CFLP. Electrophoretic method. SSCP, single strand conformation polymorphism. When a short DNA fragment is having a single base difference, it can be detected on non-denaturing gel due to the difference in conformation and hence the movement of DNA. But the technique is less reliable for larger fragments, and also it cannot differentiate between SNPs and other mutations. Chromatographic based method, DHPLC. It is used to detect DNA SNPs in less than 10 minutes following PCR amplification. Tetra-arm PCR. It employs two primer pairs to amplify respectively the two different alleles of a SNP in a single PCR reaction. A computer program designs the primer.
All of us have two alleles, right? For each gene, we all have two alleles, one from either parent, right? So in this case, it is a commercially available software which actually will design primers for you. You don't even need to do this thing. They will do the primer design, and all you need to do is use those kits for detecting the SNPs.
In this, two primer pairs amplifies to specific segments with mismatch at the 3' end of internal primer on outer forward and outer reverse, which are identical and complementary to the template. One inner forward and Inner reverse determines the allele's specificity. Primer extension, a two-step process in which a single primer which lies just upstream to the predicted SNP is used, then a mini sequencing is performed. Other methods are resequencing of genes or DNA locus using Sanger sequencing platform, and mining of sequences from public databases and aligning them to predict the SNPs by PCR validation. Applications of SNPs to detect differences between individuals or varieties at molecular level. SNPs are helpful in gene discovery, mapping, and sequencing. It is helpful in gene function identification, in plant breeding by combining traits of interest and develop new varieties, to differentiate plant varieties, useful in finding genes involved in disease and to study how the variation in DNA sequence of human respond to pathogens, chemicals, drugs, vaccines, etc. Individual identification and relationship in a family. In a forensic field, for example in DNA fingerprinting. And at last, the population or ecological studies, taxonomy, study evolutionary history of organisms, etc.
So in this case, in the pandemic, right, like this, many people responded differently. Some people had overwhelming inflammation in their lungs, right? Some people they had infection, but they managed to control the infection, right? And some people probably didn't, probably got infected and got away with it with very minor fever and all that, right? Why is it that the pathogen is same? Well, it is pathogen is common, yet all of us responded so differently, and underlying cause could simply be a SNP, in one of those genes which is involved in inflammation, right? So, in those people who had a SNP which drives inflammation, probably the disease results in severe inflammation, and people either have to be treated in the hospital, or sometimes the inflammation was so severe that it turned out to be fatal, right? And in other cases, for example, it could be that the body initiated certain protective changes, and the immune system played its own role, and the infection came under control. In certain other cases, maybe didn't really have too much of an immune response. They just had a basal immune response, and the people responded very well. So it could all boil down to having just a point mutation, or variation in that point, and then people will have different consequence.
Advantages and disadvantages. Advantages of SNPs are it provides accurate and fine gene mapping of target regions, rapid association of marker with a trait, accelerate cloning of gene of interest. SNP detection is more rapid because it is based on oligonucleotide hybridization analysis, potentially a very large number and co-dominant in nature. By tracing SNPs, it is possible to identify mutations due to single base substitutions which are otherwise non-identifiable by conventional mapping. Disadvantages. These are biallelic nature, less polymorphic, high equipment are costly, the marker development involve resequencing of even whole genome is rather costly. SNP technique is
So this process, has improved a lot. So earlier, the whole genome sequencing was costly, but now it has come down. It has come down significantly and dramatically. So in the future, it is likely that SNPs will be discovered solely based on sequencing and analysis of your DNA sequence. One of the slides was that data mining. So what is going to eventually happen is that everything is going to get stored in some place in the form of a database. Each of us, our DNA sequence at some point in time or the other might be sequenced, and you will have the sequence sitting somewhere in the cloud. All you need to do is to take, maybe few people's genome randomly and align them, and what you will see is, 99.9% identical, but also this, out of this billion, you will see literally a million base pairs, places where the sequence is going to be different. A combination, right? A combination of these differences is what results in the, identifying individuals from a crowd. Okay, and that is typically what is used for DNA fingerprinting also. Okay, let's look at another quick this thing.
You created a business. Now,
Let's watch this.
अरे नहीं चाहिए यार कोई बेमतलब का प्रोडक्ट, रखो फोन। ये प्रोडक्ट किसी काम के नहीं है, एक्ने कहीं नहीं जाते। जिंदगी के सिर्फ दो ही मायने हैं, अपने
Your nutrotherapy report packs in a ton of information, and that information is only going to keep growing. So this quick video is an explanation of some basic terms that will help you understand your report a lot better. So let's get to it. First of all,
so as you can see, the advances in medicine is going to involve sequencing, right? At some point, the sequencing is going to come down so dramatically that, every time a patient visits a doctor, it is probably going to be, as I said, people's genome will be sequenced, it will be kept in the cloud, and the doctors will be trained to look at your genetic information and also, the underlying cause for a disease, right? They might tell you the reason why you are having this disease, what could be done to alleviate the disease, if there are any. By then, it is hope that, personalized medicine will go much larger, it will occupy a much larger space, and each treatment today, for any throat infection, all of us take pretty much the same antibiotic, right? Or maybe a few different antibiotics. But I think once this, personalized medicine comes, they might say that this antibiotic is better than this antibiotic because of your genetic sequence. It is likely to come, but as he says, it does look like maybe a decade from now, the DNA sequence for all the patients will be so commonly and routinely used in a physician's lab that it may be helpful to make decisions in the clinic based on your DNA sequence.
what the heck is DNA? Think of DNA like computer code that programs your cells to make all of the proteins that your body makes. Your body uses proteins for everything. Your hormones are proteins. The receptors for the adrenaline and dopamine in your brain, those are proteins. Proteins control your entire life. And your DNA is the program that tells your cells which proteins to make and when to make them. Just so you can understand your report a little better, I need to show you how that DNA computer code is written. DNA is made up of building blocks called nucleotides. And there are 4 nucleotides: adenine, guanine, cytosine, and thymine. A, G, C, T. Your DNA is the code for everything your cells do, but it's actually the simplest computer code you'll ever see. The entire coding language is just those 4 letters, called nucleotides, shuffled around in various combinations. If you remember the movie Gattaca, it was about Ethan Hawke trying to buy Jude Law's DNA because Jude Law was engineered in a lab to have better DNA. And they made the name of the movie Gattaca using only the 4 letters A, G, C, and T. They were trying to be clever because all of our DNA is made up of only those 4 letters in various combinations. So remember those 4 letters, A, G, C, T, adenine, guanine, cytosine, thymine. How does DNA use just those 4 nucleotides to code for thousands of different proteins? The answer is in your protein shake that you probably had when you got up this morning. If you look at your protein shake, what do you see on the label? Well, you see a long list of amino acids: leucine, isoleucine, valine, methionine. There are 20 different amino acids. Proteins are just long chains of amino acids strung together in various combinations. So the way your DNA codes for proteins is by telling your cells which amino acids to stick together in what order. And it does that by using a simple code. It says, okay, I've got 4 nucleotides to choose from, A, G, C, or T. I'm going to grab any 3 of them, so AGG or GCT or AAA. Every possible combination of 3 nucleotides is a code for one of those 20 amino acids. Check out this chart. This says it all. A codon is a set of 3 nucleotides, AGG or GCT, etc. Here's a list of all 64 codons, all the possible combinations of 3 nucleotides, showing you what amino acid they code for. So this is the code for how to make all the amino acids in your protein shake. It's a super simple code, but if you stick thousands of these amino acids together, you get proteins that are infinitely complicated. So that's how proteins are made: long strings of codons, which means long strings of thousands of amino acids in any order you need. That's how you can get all of the variety of life on Earth using only those 4 nucleotides, A, G, C, T. And that's how DNA works, which brings us to the part we really care about, which is, what's a mutation? So when one of your cells divides, it has to copy all of that DNA into the new cell. And sometimes, as it's making a copy, it screws up and it sticks the wrong nucleotide in one spot. So what does that mean? Well, check this out. You can see there are actually 4 different codons that make the amino acid valine. There are 4 different ways to make valine. So if a mistake happens on the last letter of the codon, and my DNA actually swaps that T for a C, it actually makes no difference because GTT and GTC both code for the same amino acid. I'm still making valine. But now look at what happens if the mistake is in the middle nucleotide and I change GTT to GCT. Now suddenly I'm making alanine instead of valine. And if the mistake happens on the first letter and I swap a G for an A, now I'm making isoleucine instead of valine. So you can see that some mutations make a big difference and some don't. If it doesn't change the amino acid, the mutation usually makes no difference. The point of the Nutritherapy report is to look at the mutations that make a difference in the code and actually affect how we feel. So here's the last point I want to make. I used the word mutation, but often when you look at genetic reports, you'll see a different term used, either variants or SNPs, SNPs. If I have a C in a certain spot in my DNA and you have a T in that same spot, scientists call that variation a single nucleotide polymorphism, or SNP. So when we talk about your SNPs, we are talking about mutations that changed one letter in the codon. Got it? So when you see a genetic report, here's how they'll talk about it. They'll give an ID number to that particular variant called an RSID, which stands for Reference SNP ID. And they'll name the gene, and then they'll say what letter got changed, meaning what nucleotide got changed in that mutation or SNP. So for example, the COMT gene, which controls dopamine levels in your brain, has a well-studied SNP, RS4680, and some people have a G, or guanine, and some people have an A, or adenine, at that location. And because that changes which amino acid is coded for, some people make a valine, and some people make a methionine at that location. So that is often referred to as the COMT val-met SNP. And here's the last point I want to make. remember that you get one strand of DNA from your mom and one from your dad. So at that particular location, RS4680, you might have a GG or a GA or an AA. That's your genotype. And you'll see your genotype written sometimes with the letters separated by a semicolon or a forward slash or nothing at all. So now you're ready to start looking at your own genetics. Welcome to the rabbit hole.
All right. So I think this thing made it very clear, what SNPs are, how SNPs are identified, how SNPs are labeled, right? Any questions on SNPs so far? Okay, there are no questions. Let's just see one other quick video and then we'll
Create videos for your business.
Hello and welcome to AJ Science Series.
This is a short video, but the audio is kind of
Single
faint.
nucleotide polymorphism. Human genome consists about 3 billion base pairs in the form of DNA. DNA sequence is formed of a chain of 4 nucleotide bases: adenine, guanine, cytosine, and thymine. Double-stranded DNA is bind due to complementarity between adenine and thymine, guanine and cytosine. DNA is the code book for how each individual form appear, grow, and operates. The genome is organized in 23 paired chromosomes. To understand single nucleotide polymorphism, let's see an example. In a video game, if a child types CAT, his cute cat picture appears on the screen, and he enjoys this, and he does this again and again and again and again, and while typing CAT by mistake, in place of C he types R, and the picture changes to mouse. The same thing happened in single nucleotide polymorphism, in which the change is the nucleotide, single nucleotide change occurs. Similarly, in human genome, single nucleotide polymorphism are the most common types of genetic variation, and which represents a difference in a single DNA building block called nucleotide. For example, single nucleotide polymorphism may replace nucleotide cytosine with nucleotide thymine in a certain stretch of DNA, and maybe this could be the cause of your eye color, which may be different from your friend. Two important genes responsible for eye color are KERG2 and OCA2, and due to single nucleotide polymorphism, different eye colors are present in human population. SNPs are found throughout the entire chromosome, including promoter, exon, introns. However, 5% DNA code for proteins, and most SNPs are found in the non-coding regions. They occur in every 300 nucleotide on an average, which means that there are roughly 10 million SNPs are in human genome. If they occur in coding region, then they could cause different expression if they code for different amino acid. And for example, a specific SNP in sickle cell anemia, which causes red blood cells to be in the shape or look like a crescent or sickle, and is caused by just one substitution to the codon GAG, which codes for glutamine, and in GAG the adenine is replaced by thymine, and in GTG code for valine, thus exactly one SNP at just right place in the genome can cause sickle cell anemia. So the question arises in our mind, why SNPs are important, why we need to search for SNPs, and why so much research is going on SNPs. Most SNPs have no effect on health or development. They are commonly found in the DNA, but they can act as biological markers, helping scientists to locate genes that are associated with diseases. Researchers have found SNPs that may help to predict an individual response to certain drugs, susceptibility to environmental factors, and risk of developing particular diseases. Particular SNPs are detected by comparing regions of genome between cohorts. So SNPs act as chromosomal tags to specific regions, and these regions can be scanned for variations that may be involved in human genetics and disorders.
There is a new branch of medicine or rather pharmacy called pharmacogenomics, right? Based on your sequence, how do you respond to medicines, right? Along with personal genomics, your sequence will also tell you how you are going to respond to a medicine. For example, you can give morphine to certain patients and they will have absolutely no effect, right? They don't feel any reduction in the pain. And there are certain other people for whom you can give an extremely low dose of morphine and they get normal. They are relieved of their pain. And then you have another group of people who are neither of those, so you'll have to give them a little higher dose. And all of this is simply because of the changes in the way morphine gets metabolized in the people's body, right? Some people are called extensive metabolizers and some people are called as poor metabolizer. So if depending on the parent, one parent could be an extensive metabolizer, one parent could be a poor metabolizer. What will happen is the progeny will be intermediate metabolizer. So for a poor metabolizer, you need to give them morphine will not have any effect. For an extensive metabolizer, morphine will have an effect even at a very small dose. And for an intermediate metabolizer, you may have to give them a dose that is little more than extensive metabolizers. Okay, these are all I just want to tell you is that these are very very diverse. This thing, the SNPs can have very very diverse effects. Okay, similarly there are some medications given for heart patients, and depending on the sequence, your genetic sequence. Of course, doctors at this point in time, they don't know what your genetic sequence is, right? So they will give you an extremely low dose and then start incremental dosages. They'll keep on increasing until the patient feels free, better. Okay. But the moment your genome is available and the doctors are in a position to take a call on, based on what your genomic sequence is, how you are going to respond, it will become truly a case of personalized medicine. Okay. Similarly is the case for vaccination. Some people are able to mount a vigorous immune response for in the current COVID pandemic. On the other hand, some people are not able to mount a vigorous response, right? Maybe those people who are not able to mount a vigorous response, they may have to be given, additional booster doses. Okay, that need not be the case every this thing, but at least for certain people, it is advisable. So it is we are looking at a day when sequencing will be so common, and you yourself will be able to figure out what is best for you. Of course, it's going to take time and effort on your part, to understand how you are going to respond to anything, right? Whether it is a pathogen, whether it's a vaccine, whether it's a drug, it's going to take some more time. Okay. So this is what I wanted to talk about SNPs. Any questions on SNPs? All right. So,
So, share the
yeah,
links of the video.
what is that?
So, am I audible?
Yeah.
Sir, can you just share the links of the
I
videos
will,
that
I
you
will, I
showed?
will. Sure, sure, sure.
Okay.
I will do it. Okay, thank you. I'll see. I should have some notes. I'll probably also share the notes. Okay.
Okay, sir.
All
Thank
right.
you.
So we will meet in tomorrow's class where we will start looking at the applications. The real applications, how transgenic technology introduce foreign DNA, what all the methodologies are available, and all that. Okay, we'll see in tomorrow's class. Okay. So take care.
Thank you, sir. Thank you, sir.
Yeah.